Good day, everyone. I am Maria Cristina Ablao. I am one of the reporters of Chapter 18, Techniques in Cell and Molecular Biology. I am tasked to discuss from topics 18.1 until 18.6. First off, let us discuss topic 18.1, the light microscope. I know that we are all very familiar with what light microscope is because I believe that we've already tackled this one during our high school biology class and even during grade school, 'di ba? We were already taught about the parts and functions of microscope. Today, let us just do a recap. We'll dig deeper about this topic. So, are you guys excited? All right. So, what is a light microscope? Light microscope is an instrument that uses visible light and magnifying lenses to examine small objects not visible to the naked eye or in finer detail than the naked eye allows. So, microscopes are basically instruments that produce an enlarged image of an object. So, this is how light microscope or optic microscope looks like, no? As you can see on the illustration. So, the two diagrams or illustration that you can see on your screen, do not just be confused, all right? These two are just the same. So the diagram on the left side of your screen, I got this one from our main textbook, and the diagram on the right side, I got this one from the internet. And I just specifically included this one because it includes the general or the entire parts of a light microscope. So, para mas maklaro na to. So, how does a light microscope operates? So, here we have the light source. Alright, I hope makita ninyo ni na cursor. So, light source is the one who provides light for viewing the specimen. Now, when we say specimen, this is the slide. So, this is the object being examined. So, light passes through a lens which focuses the light onto the specimen. After passing through the specimen, the light passes through the objective lens, which magnifies the image from 4 to 100 times its original size. Next, the light travels through the tube of the microscope and the eyepiece, which further magnifies the image by a factor of 10 or 15 before it finally enters the eye. Also, it is important to remember that some light microscope can magnify an image up to 1,200 times its original size. So it's very amazing, 'di ba? So the stage of the microscope is placed for observation. Stage clip is designed for the specimen to be in place. So para intact. Attached to the stage is the diaphragm, which controls the contrast of the image by controlling the amount of light from the light source that you expose the specimen to. On the sides of the stage are these slide adjustment knobs, which are used to move the specimen so that what you want to see is in your field of view. Now, above the stage are the objective lens that are held by a nose piece, which can be rotated so that different objective magnifications may be employed to view the specimen. The tube of the microscope connects the nosepiece to the eyepiece, which further magnifies the image and is the part one looks through to view the specimen. There are also focus knobs on the side of the microscope, the larger one being the quartz focus knob. So here it is. This one is used for larger changes in focus. We also have the smaller one being the fine focus knob. Here. This is used for smaller adjustments in focus. Now the rheostat, located here at the bottom. Alright. This is used to turn the microscope on before use. Now, since we have discussed about the parts and functions of microscope, let us proceed to the other slide. Now, there are important parameters in microscopy that we need to understand. We have the magnification, resolution, and contrast. So when we say magnification, this is the ability to make small objects seem larger, such as making a microscopic organism visible. Magnification is the process of enlarging something only in appearance and not in physical size. Resolution, on the other note, is the ability to distinguish two objects from each other. And contrast is the difference in light intensity between the image and the adjacent background relative to the overall background intensity. Modern light microscope use several different modes of operation depending on the needs of the investigator. So there are various types of light microscope. We have bright field light microscope, phase contrast light microscope, dark field light microscope, and fluorescence light microscope. First, let us discuss about bright field light microscope. So, bright field light microscope is one of the primary and favorite techniques used in all forms of optical microscopy. Now let us tackle about the preparation of specimens for bright field light microscopy. Specimens to be observed with a light microscope are broadly divided into two categories. We have whole mounts and sections. A whole mount is an intact object, either living or dead, and can consist of an entire microscopic organism such as a protozoan or a small part of a larger organism. Most tissues of plants and animals are much too opaque for microscopic analysis unless examined as a very thin slice or section. So when we say opaque, this is defined as not transparent or not allowing light to pass. So it makes it hard for the scientist or the researcher to study or understand the content of an organism. Now, to prepare a section, the cells are first killed by immersing the tissue in a chemical solution called fixative. A good fixative rapidly penetrates the cell membrane and immobilizes all of its macromolecular material so that the structure of the cell is maintained as close as possible to that of its living state. The most common fixatives for light microscope are solutions of formaldehyde, alcohol, or acetic acid. Now, after fixation, the tissue is dehydrated by transfer through a series of alcohols and then usually embedded in paraffin or wax, which provides mechanical support during sectioning. Paraffin, by the way, is used as an embedding medium because it is readily dissolved by organic solvents. Slides containing adherent paraffin sections are immersed in toluene, which dissolves the wax, leaving the thin slice of tissue attached to the slide where it can be stained or treated with antibodies or other agents. After staining, a cover slip is mounted over the tissue using a mounting medium that has the same refractive index as the glass slide and cover slip. Now let us proceed with phase contrast microscopy. Phase contrast microscopy was first described by the Dutchman Frits Zernike in 1934. This technique is ideal for thin, unstained specimens which are approximately 5 micrometers thick above the nucleus but less than a micrometer thick at the periphery. Small unstained specimens, such as a living cell, can be very difficult to see with a bright field microscope. The phase contrast microscope makes highly transparent objects more visible. Phase contrast microscopes accomplish this result by first separating the direct light that enters the objective lens from the diffracted light emanating from the specimen, and second, causing light rays from these two sources to interfere with one another. Phase contrast microscopes are most useful for examining intracellular components of living cells at relatively high resolution. For example, the dynamic motility of mitochondria, mitotic chromosomes, and vacuoles can be followed and filmed with these optics. The greatest benefit derived from the invention of the phase contrast microscope has not been in the discovery of new structures, but in its everyday use in research and teaching labs for observing cells in a more revealing way. The phase contrast microscope has optical handicaps that result in loss of resolution and the image suffers from interfering halos and shading produced where sharp changes in refractive index occur. The phase contrast microscope is a type of interference microscope. Now, when we say interference microscope, it is one of the techniques used to visualize the surface features. So the illustration that you can see on the screen, so that is a phase contrast microscope. Now, let's proceed with dark field light microscope. In this technique, the condenser directs a cone of light onto the specimen at high azimuths so that first-order wave fronts do not directly enter the objective front lens elements. Light passing through the specimen is diffracted, reflected, and refracted by optical discontinuities, enabling these faint rays to enter the objective. The specimen can then be visualized as a bright objective on an otherwise black background. Dark field microscopy is a specialized illumination technique that capitalizes on oblique illumination to enhance contrast in specimens that are not imaged well under normal bright field illumination conditions. So, dark field microscopy is an excellent tool for biological and medical investigations. It can be effectively used at high magnifications to image living bacteria or at low magnifications to view and image cells, tissues, and whole mounts. Now let us discuss fluorescence light microscope. The fluorescence microscope allows viewers to observe the location of certain molecules called fluorophores rather, or fluorochromes. Fluorophores absorb invisible ultraviolet radiation and release a portion of the energy in the longer visible wavelengths, a phenomenon called fluorescence. The light source in a fluorescence microscope produces a beam of ultraviolet light that travels through a filter which blocks all wavelengths except that which is capable of exciting the fluorophores. So here are sample images taken by various types of light microscope. No? So as you can see on the illustration, there is a image that was observed or taken by bright field microscope, phase contrast microscope, and dark field microscope. Before we will hop on to another lesson, let us discuss about the importance of light microscope. Light microscopes play an important role in many research laboratories, including electron microscopy facilities. They can be used as a primary visualization tool or in support of electron microscopy. So generally, light microscopes is very important.